Hi there. In this video, we're going to walk through Pluto Spatial Studio. So we know that spatial data is of interest to many scientists, so we set up this sandbox environment with 20 published datasets where you can go in and explore the data and really get a feel for Spatial Studio. You'll notice that these datasets span lots of data types like Visium, Xenium, Merfish, and more. For this example, I'm going to talk through human breast cancer Xenium panel. So right away, you'll notice that there are quite a lot of analyses here, and you can actually change this layout to be specific to your scientific question, whether that's differential expression or co-expression or gene analysis. But starting at the highest level with tissue morphology, in this left-hand panel, you can completely remove the spot sets and scroll around and zoom in to view regions of the tissue in very high resolution. You can also see the tissue colored based on cluster, so you can see that there's this fatty acid synthetase enriched region, as well as this pink TROP2 enriched region. And then often the natural next question is to compare those regions and identify differentially expressed genes. And to do that, we can use our lasso tool. So I'm going to lasso this blue fatty acid synthetase enriched region. And then I'm also going to lasso this this region that maintains this glandular epithelial structure. And then run differential expression. And what that returns to me is a volcano plot showing the genes that are significantly differentially expressed between group A and group B, but it also shows gene set enrichment between those two regions as well. So you can see GO gene sets as well as KEGG and IMSIG-DB. You may also be interested in exploring individual gene expression or co-expression. So we can switch the layout to look at gene analysis. And then you can see this gene, which is selected called LUM, visualized on a map. And then you can also see the dot plot here showing all of these genes and their expression across all the clusters, as well as a violin plot of LUM here. And then you can add a second gene to that and look at co-expression. So let's say that we want to look at EpCAM as well as LUM. And we'll see LUM colored in green and EpCAM in red here. So you can see that visualized on the tissue map. And then you can also see the relationship between the two genes in this co-expression plot as well. And so hopefully that was a helpful quick walkthrough of some of the features in Spatial Studio. If you're interested in learning more, I'd love to schedule a demo with you and we can dive into the data together. Thanks. Bye.